Okay, so today we're going to look mostly at muscle physiology today. Last time we talked about all of the protein filaments that we find inside of the sarcomere, and today we'll talk about how they work and what they do, and how the whole what's called the sliding filament theory comes together. And so yeah, so this again we'll kind of review those key proteins that we talked about last time. Okay, so the key players here in the sliding filament theory would be the first one that's called the heavy chain, which is myosin. Okay, this is the one that has the head attached to it, and so the function of this is the one that's actually going to move actin. Okay, this is the one that's going to cause the sliding. It's not the one that actually slides, but it's the one that causes the sliding. Okay, and then we have the light chain, which is called actin, and that's the one that actually slides. It gets moved by the myosin head. Okay, and then we have tropomyosin, which its role is to kind of cover up the binding site for where that myosin head is going to attach to actin. So it kind of gets in the way of that. Okay, then we have troponin. If you looked at last time, this is the one that has a binding site for calcium. And when calcium binds to it, it'll change its shape completely. And then that helps to push tropomyosin out of the way. So then we can open up that binding site for the myosin head to bind to actin. And calcium's a key player here. Calcium's going to bind to troponin and cause its shape to change. And then ATP, adenosine triphosphate, its function is to provide power to this whole situation, but not necessarily at the time that we would think it would provide power. We would probably think that the power would be used or the energy would be used when the sliding actually happens, but it's actually before that. It's when we cock the myosin head, kind of put it into its power-packed position. Okay, so take a sec here. I think the colors are somewhat reflective of what those words are there. They're a little bit tricky, but decide and chat with people on your team for a sec which one there is tropomyosin and send that in on your clickers. Sorry to any students with color blindness. It's probably not a good slide for you. But kicked your water bottle over there, Maddie. I'm sorry. All right, go ahead and send that in if you haven't yet. What'd you say?
All the purple was pink.
I know.
Totally.
I know. Becomes more of a question about that than anything else. All right, anybody else? I got 41, 42. There we go, 43. Okay, so about 2/3 of you, 68% are saying B. Okay, so the blue one. Yeah. And so the blue one would be, if those of you are wondering about the color that I'm talking about, it's is a moving image, which makes it harder to point at things. But yeah, so the like long string like blue one there, right? That's the one that would be tropomyosin. It is kind of a long strand that covers up a portion of the actin, right? Yeah. And so the red one there is the myosin with the head, right? Yeah, the kind of the green and yeah, the green and that like cone shaped gray one that comes in would be the ATP. The little yellow dot would be the calcium. That's that little yellow like ball that comes in and then pops back out. And then and then we can see the kind of orangish, yellowish beads that are all strung together. That's actin, right? And then troponin is the one that looks like a little mini snowman kind of, but the pinkish one. Yeah. So yeah, so that's that. Questions on any of that? So we'll talk about how all this comes together here. Okay, So we're going to talk about two primary processes here, and the first one is called excitation contraction coupling. Okay, so this is going to start with an action potential in a neuron. So an action potential is a nerve signal. That's like the communication part of the nervous system. That's the signal that goes down an axon, travels to the target tissue. That's an action potential. So it starts there, and then it ends with calcium binding to troponin. Okay, so we saw a lot of that happening in that little GIF on the last slide, but we saw the calcium binding to troponin, and then the part that actually produces force or creates force is the cross-bridge cycling part, which would be the cycle of the myosin head cocking, attaching to actin, pulling it back, coming off, cocking again, attaching to actin, pulling it back. It's just a cyclical process that happens over and over again in a matter of milliseconds, which is happening so many times, even just as I'm talking, right? All my muscles in my neck and head and all kinds of things are doing that literally millions and millions of times just to produce any kind of movement that happens in our body. So, all right, so let's look at those. We'll start with the excitation contraction coupling part first. I don't think this is going to work on my iPad, but why not give it a shot? We'll stop recording here just so I don't have any YouTube violations. Somebody else's YouTube. Good to see you. Yeah, I'm doing good. Let's talk after class for a second about that. The big yellow thing, yep.
I have a question.
Sure.
So when ATP binds, how will the calcium be gone or does the calcium stay? Or does it get?
Calcium stays there, yeah.
So,
Yeah, that video kind of made it sound like a new calcium comes or something. We don't have to have new calcium. It can just stay there.
so just when the ATP comes in, that's when it stops. How would it know like when to stop?
What do you mean, stop it?
Halt.
Like stop
Like-
contracting?
Yeah.
Yeah, so that would be when the sarcoplasmic reticulum starts sucking the calcium back up. And so then there's a low concentration of calcium around the myosin head, so then it comes off of the myosin head. So yeah, it'll come off of the troponin because if there's not a lot of calcium around it, kind of just like a diffusion type thing, you know. Mhm. And the reason that we would start, you know, that would happen would be because we're stopped sending a nerve signal. So we stop getting the signal to release calcium from the sarcoplasmic reticulum. So then it just starts going back the other direction.
Action potential is what indicates how much calcium is flowing in.
Pretty much like, so the frequency of action potentials, so there's gonna be a bunch of action potentials coming down and that causes the calcium to be released. So if those slow down, then not as much calcium is released, something gets pulled back up into the sarcoplasmic reticulum. Yeah,
Okay,
so, yeah.
thank you.
Mhm.
So it says that it rises at the neuromuscular junction.
Yep.
So
That's the combo of the nerve, the space between the two, and the muscle cell. This is all the neuromuscular junction. This whole thing right here. Just that part, yep.
and then is this a close?
That's the T-tubule, uh-huh. That's just zoomed in. You can't see you can kind of see the T-tubule there, but yeah. Mhm. Okay.
So when this turns into ATP and it's like connected right here,
Yeah,
does it go and connect to like the next one or does it just do the same one over and over?
yeah, so, yeah, and that's part of the cross-bridge cycling. We'll get to that part, but pretty much after it pulls it, then it detaches
Just-
and then gonna go to like the next little dot there.
Okay,
Or, yeah, or a couple down.
and then
And that's kind of what she's there. So it'll grab the next one and then pull it. So it's kind of like it like ratchets down further and
what
further.
happens when, like it's right here?
Yeah, then it can't shorten anymore. So then that sarcomere can't help us produce force anymore. That you're
So that's
getting
like the
at.
most our muscle can like stretch or like contract.
Yeah, kind of, but this is like very microscopic, right? So we have millions of these sarcomeres inside of each muscle. So when we're right here with our biceps, some of them are already all the way shortened and we're not using them anymore. But then some of them still have some room to shorten, kind of, they're all in different positions. And so yeah, but we'll get to that soon when we talk about like force production in muscle and it. Turns out that you can't produce as much force right here as you could like right here in the middle. And it's because a lot of those are already fully shortened and they're not helping us anymore. So, yeah. So you're writing in, discussing what's happening in each image there. You're putting your phones in your backpack. You're staying engaged. Talking you through what I want you to do. They're not on your desk because they distract us, even if they're just sitting there. Yeah.
So then. I think action potentially is like plus and minus.
Yeah, that's kind of represented by that. We'll get into the details of that more, but the green line kind of represents it too. But yeah, yeah.
But I can't tell until it's zoomed in on the bottom part that it's like that. Yeah.
Yeah.
All right, let's go ahead and come back together here. And we'll kind of walk through this, okay? So let's look at this first image here. So just to kind of orient ourselves, this is a neuron, right? What we would call a motor neuron, which just means that it only carries signals coming from the central nervous system out to the peripheral nervous system, and it's going to activate a muscle cell for us. Okay, and so that's our neuron. So we got this signal traveling down it. This signal we'll just abbreviate as AP, but that's an action potential. Okay, so an action potential travels down the neuron there, and bottom line is it is going to create another action potential on the muscle cell. Okay, we're skipping a bunch of steps in here in the middle, but we'll get to that in another in another day of class. Okay. But as far as the steps say here, what does it say? It just says motor neuron sends a signal to the muscle, arrives at neuromuscular junction, it starts an action potential, right? So we're seeing all 1, 2, and 3 here. Okay. The neuromuscular junction is this part right here. So NMJ, neuromuscular junction. It includes 3 components. It includes the end of the neuron, which is the yellow part there. It includes the space between the 2 cells, which is just right in here, okay? And then it includes the special part of the muscle cell membrane that has a bunch of receptors for the neurotransmitter there, okay? And yeah, what did we call the muscle cell membrane again? What was that special name for it? Sarcolemma, there we go, yeah. So the sarcolemma is a special cell membrane. It's the one that we find on muscle cells, okay? So we got the signal going down the motor neuron, causes an action potential on the muscle cell. That's pretty much all we got here so far. Okay. Questions on that one at all? Anything else you see in there? Okay. All right. So then we know that muscle cells are super thick. They're way too big to just allow the signal to stay on the outside of the cell and have an effect. So we need a special structure that lets that action potential dive deep into the muscle cell. And that's what those T-tubules do for us, right? So those T-tubules allow that to happen. So the action potential, the action potential goes down the T-tubules. Okay. And then we go to the next image. And we can see that those T tubules are directly connected to little membrane proteins that are embedded in that blue thing. What's that blue thing? Perfect. Sarcoplasmic reticulum. Okay. And inside the sarcoplasmic reticulum is where we find a bunch of calcium. Right. So the sarcoplasmic reticulum stores calcium for us. And so then we have ion channels here that are voltage gated ion channels, meaning that we change the voltage on the membrane and the door opens up, right? And so that's what happens. So we open up the door for calcium to flow out of the sarcoplasmic reticulum. And you can kind of see in the background here some actin and some myosin. So that's what's happening. This, all that calcium is then flowing into sarcomeres that are just right there in the background. Okay. So action potential stimulates opening of calcium channels in the SR, or that sarcoplasmic reticulum. Okay, so calcium flows out of the sarcoplasmic reticulum. Okay, got that? Questions on any of that? Okay, and then what does it do? Now we're zooming in closer. So we're seeing here, right? So this would be this one up here is like a closed calcium channel on the sarcoplasmic reticulum before the action potential gets there. And then we can see this one as the action potential comes down, it opens up that calcium channel, calcium flows out, and then we zoom in right here on a little sarcomere that's behind, right? And we see on the left side without calcium, troponin is there. And you can see tropomyosin is covering up those little binding sites on the actin molecule, right? And then calcium comes and binds the troponin, changes its shape, which then it pulls tropomyosin along with it. And now we've got open binding sites for the myosin head. Okay, so that's what's happening. We're just opening up those binding sites there. Okay, and this takes us back to when we talked about proteins. We just talked about proteins a little bit in this class, but remember the different levels of structure, right? We had primary structure of a protein, secondary, tertiary, quaternary. The primary structure was all covalent bonds, peptide bonds, right? Really strong covalent bonds. But then once we get above that to secondary and tertiary and quaternary, then we start kind of relying on some hydrogen bonds. We rely on the way that the amino acids next to each other interact with each other. So for example, we might have an amino acid, two amino acids right next to each other where they're both positively charged, right? So we got that peptide bond that's holding them together, both positively charged. They're going to push apart from each other like this because of having like charges, right? Maybe we have one here that's positive, one here that's negative. Those would bend towards each other. This is all what creates the shape of the protein. So we might have that in a couple of those amino acids in troponin, but then calcium comes and binds to that negatively charged one, right? And then it totally changes the charge of that one. So then it folds back. Instead of folding this way, now it folds this way, right? That's just a small example, but that's the kind of thing that changes the shape of troponin, right? We just change the characteristic of one amino acid that is in troponin by binding calcium to it, totally changes its the shape of that protein, which is what enables us to do this process. All right, we'll see the same thing with the myosin head in a second. When phosphate binds to it, when we break down that ATP and bind phosphate to it, it totally changes the shape of that myosin head, which is the same mechanism that allows everything in biology to move, right? Things, all animals, all plants that can move around a little bit, they do it with some form of myosin conformational change in the protein structure because that phosphate binds to it, right? And so it's this beautiful situation where it's just a single amino acid, like in the right place that has that binding site for one of these things, and it totally changes the shape and is what allows, which activates all of life in biology is this type of movement. So yeah, so that's the excitation contraction coupling part. Questions about this before we go on to the next part? Okay, so then we get to the cross-bridge cycling part, which is kind of the part where it's going to go over and over again. And this is what is actually moving the actin and the myosin. Okay, so the first thing is this reaction called ATP hydrolysis. Okay, that's just the breakdown of ATP, turning it from ATP into ADP and phosphate. Remember, ATP is like it's a nucleic acid, right? So meaning that it has a base on it, one of those nitrogenous bases, and then it has some phosphate, right? And yeah, and so then which one is it more like? Remember, we had DNA and RNA when we talked about nucleic acids. Which one was ATP more like? You guys remember? Yeah, so I don't have a good chemical image of it here, but it's a little more like RNA because it has that ribose as its sugar, right? So I'll have a sugar, a phosphate group, and the nitrogenous base. But just like how DNA and RNA have the C or the G, right? The A, the T, or the U, right? It's always the A one with ATP, right? So it's just always the A one, the adenine. Okay, and so, but instead of having one phosphate, it has three, right? That's the unique part about ATP. And then those phosphates are all kind of bound to each other, but somewhat unstably, right? Meaning that last phosphate on the end kind of wants to bind to something else. And when it does bind to something else, it exerts power on that. And that's the way we do all chemical reactions in the body for the most part that require energy is via ATP, is via taking that last phosphate and binding it to something. And then it totally changes the shape of that thing or activates another process. Okay, so this is our chemical energy. So doing that, using the power in ATP provides for the cocking of the myosin head, putting it into the power pack position, but it does this before it attaches to actin. Right? See that it's not even attached to actin yet. So meaning that when the actin actually slides, that's after we've used ATP, right? We use the ATP to cock the myosin head, then it attaches, okay? And then the part where it actually slides, this is actually the sliding part. This is when our muscle shortens, is during the power stroke. This happens when the phosphate comes off of ATP, or sorry, comes off of the myosin head. Okay, this says with the release of ADP and phosphate off of that myosin head, then the myosin head goes back to its original shape, but it pulls actin along with it. Okay, so it's attached to actin, goes back to its shape, and it pulls actin along with it. That is what the shortening is inside of our muscle. Okay, and then the myosin is just stuck to the actin there. And the only way it comes off is if a new ATP comes, and then we go through that process again. Okay, but we have to have a new ATP molecule to get that myosin head to detach. And then we break down the ATP again, cock it, attach it again, power stroke again, just over and over and over again. Okay. So when somebody dies and they stop breathing, they stop producing ATP, right? They can't make ATP anymore if they don't have oxygen coming in. And so what happens to all of your muscle? All of your muscle has the myosin head attached to actin and it's just stuck like that. Okay. Because the thing that allows number 4 to happen again is the new ATP, right? So if there's no new ATP coming, then the myosin heads are just stuck on actin. That's rigor mortis, right? That's why people go into rigor mortis, is because of that myosin head being stuck. Okay, questions about any of that so far? Okay, so same thing here. There's a series of 5 slides with images. Just walk through these with the people on your team, kind of fill in, discuss what's going on, and then we'll come back together here in a couple minutes and make sure we're all on the same page.
Sounds like this, my head is my. Yeah, right?
What's that?
Like, this is my head into
Oh,
position.
oh, there you go.
And
Yeah,
then it connects.
I like it. I like it. I always tend to use my arm like this, but this is more interesting for sure. Yeah. I like it. It's a full body interaction with the material. I like it.
Like taking a test.
Yeah. I wasn't sure if you were like falling asleep or what was going on. Yeah, people get up doing all kinds of moves.
You're gonna put some
Yeah.
glass like this.
Yeah.
If you put a question about like this one and then everyone's.
Oh yeah, for sure. Yeah. You can count on it.
And it's like all over it.
Yeah, and then what you guys can do on your clickers here is this last slide has a clicker question. So go ahead and you can answer that one once you get there.
Yeah. I see.
All right, go ahead and send in the answer here to this clicker question if you haven't yet. All right, well, let's come back together and we'll come back to that question here in a sec. All right, you guys ready? Okay, so let's walk through these steps here. So here's our primary steps, and then these are just kind of the images of that, okay? So again, step number one here, we can see ATP becoming ADP and phosphate, okay? So we see the breakdown of ATP happening. We see the hydrolysis of ATP happening, okay? And then we can see by doing that, it's really the binding of this phosphate to the myosin head that causes it to just to completely change shape, to go from being kind of in this little bit less than a 90-degree angle, like a 45-degree angle, up to a 90-degree angle, right? And again, it's not attached at that point, right? There's no attachment yet at that point. So the use of ATP is used to cock the myosin head to put it into what's called the power pack position, okay? So we got that. Step two here, now the myosin head goes up and attaches to actin, okay? So it just moves upward, attaches there, and still there. The phosphate's still connected. Everything's the same except for it just attached to actin. Okay, that's all that happened in step two. Step three now is phosphate and ADP come off. Okay, they're removed from the myosin head, and then that makes the myosin head go back to its original shape, back to that 45-degree angle. And since it's attached to actin, it just brings actin with it, right? So it just pulls it back. This is actual muscle shortening right here. Right, so when I contract my bicep to lift up like this, go into elbow flexion, that's what's happening a bunch of times, right? Over and over and over again. That myosin head is pulling on the actin, and that's coming off and attaching and pulling again, but doing it super, super rapidly. Okay. And then lastly, we get our new ATP, right? So new ATP here, that's going to allow this myosin head to be removed from actin. Okay, and then if we want to continue the cycle, we'd go back to slide 1. And then again, that ATP would turn into ADP and phosphate. We'd cock the head again and just start right back over again. Okay. All right, and then we see it here kind of laid out for us. Okay, so all these different steps here in kind of a cyclical order here. So this has A, it's not the exact same order as the other one. This has A with the, what actually, you guys talk about that with your team for a second. What is happening there in A? Talk about that with each other for just a sec. Which step would that be from the previous ones? Okay, what do we call A there? What is that? Power stroke. Yep, absolutely. So that is the power stroke. All right, that is muscle shortening right there. Jeez. Yeah, so then we get the new ATP coming here to pull us off, right? Then that ATP breaks down and the myosin head cocks in C, okay, which was the same as step 1 there, right? Which is what most of you put on the clickers. 82% of you said that, okay? And then D would be that myosin head attaching to the actin once it's got put into that cocked or power pack position. Okay. Questions on that? Go ahead. Cramp? Yeah. So cramping, can be a bunch of different things. A lot of it has to do with kind of, it would be this for sure. It would be that your muscles are forcefully shortening, kind of, but just involuntarily. A lot of times because there's some stuff happening at the neuromuscular junction. So if we go back to this part, if there's some like fatigue up here, some too much calcium building up in this area, too many hydrogen ions building up in that area, then we can just kind of falsely activate the muscle. And so that can cause our muscle to just kind of contract even when we're not trying to get it to contract. So that's, and a lot of that has to do with ion balance and other stuff like that. So that's the easy answer, but we don't have a super clear answer of like fatigue and cramping, but that's one of the most common reasons. Yeah. So yeah, Summer. Uh-huh. EC is excitation contraction coupling, which is if we look at this slide here, which includes all of this, then that includes just this top part. Yeah, so this is excitation contraction coupling, which is pretty much preparing for contraction. I guess we need to include this part here too, because that is where calcium binds to it. That's the last part of excitation contraction coupling. And then cross bridge cycling would be that part, the CB part. And so as long as we keep sending a nerve signal down to the muscle, we'll keep pumping calcium out of that sarcoplasmic reticulum, and this cycle will continue. Okay, but as soon as we slow down that signal from the nervous system, then we stop releasing so much calcium. Calcium starts getting sucked back up into the sarcoplasmic reticulum, and then our muscle starts relaxing. Okay. All right. Other questions about that? Okay, so then we're going to switch gears here and talk about something similar, definitely about contraction, but in a different way. And so part of the this is the one place in the class where lecture and lab like really overlap each other quite a bit, right? Because you didn't have to learn the bone names in class part. That wasn't on the test, but you did in lecture or in lab. Here you have to know the muscle names in lab and be able to identify them, and in lecture too. And then in lecture we'll add the functional part of that, right? So you have to identify them, but then also know what function they do, right? In lab, you don't have to know the function of the muscles really, except for the practical, at least you don't have to, right? You just have to be able to identify them. But in class, we're looking at the naming them, identifying them, and knowing the function of them, like we did last week with the hip muscles. Okay, so one application of that is looking at what muscles activate and are used when we take a step. Okay, and so that's what we'll do just for the last part here. So when we take a step, we need to think about both legs. So one leg will be our weight-bearing leg. Okay, so as we're going to take a step, we're gonna swing one leg forward, right? One leg's gonna come forward. So the one we're talking about here is the one that's still planted. Okay, so my weight-bearing leg. Okay. The main thing that has to happen is our pelvis needs to stay stabilized. Okay, so as soon as I lift my right foot up, that automatically puts like stress on my left hip to hold me in place, right? If I wasn't contracting certain muscles in my left hip, then my hip would fall down like that, right? And so as soon as I lift my leg up, I have to contract some muscles over here to hold myself up. And the main ones would be our abductors of our hip, our abductors, right? Because the opposite of that would be adduction, which is what would happen if we would kind of fall down and let that relax. So we talked about some of those abductors last time. And the big one we talked about was the gluteus medius muscle. Okay, so we definitely need to contract our gluteus medius muscle in the leg that's stabilizing us, the weight-bearing leg, as the other leg is swinging forward. Okay, if we don't, then our pelvis falls to the side that our leg is lifted up on, and we're not stabilized. Okay, so that's the simple one. Okay, and then the other one. So now the leg that is coming forward. Okay, so we've got a bunch of things going on here, and we'll look at two different phases of this. Okay, so the first part of this will be, I'm going to take a step with my right leg. And if you want to turn around or stand up so you can see, I know it's not the best situation. I could try to stand up there on that table again. Yeah, might as well. Let's do it. All right. Okay. All right. That's better. Yeah. Okay. So I'm going to step forward with my right leg, right? But first of all, is going to be a push off there, right? So that's the first part, is the push off. Okay. So that's what we're talking about right here, the push off phase. Okay. And so we want to think about our ankle, our hip, and our knee, and what those joints are doing. Okay. So in that push off phase, our ankle is going to plantar flex. Okay, so that's pushing down like that, right? Our hip is going to extend a little bit, and our knee is going to flex a little bit. That part is kind of tricky and kind of slight, but those are the three that we're going with. Okay, and then I'm going to swing my whole leg forward like this, right? So that's hip flexion, right? My knee is then going to extend, so it's going to go from this to this, so my knee's extending, right? And if I don't lift my toes up, I'm going to trip on my toes, right? So I have to dorsiflex my ankle, right? So I got to pull up my ankle, okay? So you guys, for a sec, with the people on your team, see if you can think about which muscles are going to be used in each of those, okay? So go ahead and just talk about each of those, plantar flexion, and which ones are those, okay? Walk through that, and then we'll come back together here and make sure we got it.
It's not it. Yeah.
All right, let's come back together. One of the ones that's super tricky here is the knee. And that's because we're oversimplifying things by turning this into two stages, okay? If some of you are wondering, like, how does my knee flexing? I'm not sure if that's really, it's because the push-off phase is actually like three different phases, and we're kind of clumping it into one. We're just choosing the knee flexion part of it, which does happen just a little bit when we first push off. Our knee flexes a little bit before it extends, so don't get too hung up on that. Just believe me and go with it there. But yeah, so what are our plantar flexor muscles in our lower leg? Which ones? Gastrocnemius, yeah, and we haven't really gone into these yet, so it's okay. We have in the cat, but not, and you've seen them in the human, but we haven't talked about the function of them yet in class. Yeah, so those would be our calf muscles, right? Our gastrocnemius and our soleus. Okay. Hip extensors, so those are the ones making our hip go back. Which ones do that? We did talk about those last week. Glutes, yeah, our glutes do that, and our hamstrings do it too, right? So we'll talk about the hamstrings here. The glutes are really more active when we're in a lot of hip flexion already, meaning like if I'm going up a step like this going up, then my glutes are really more active. Where we're just kind of pushing off a little bit, it's going to be more hamstrings that do that. Okay. So all the way on the outsides, biceps femoris, then we go semimembranosus, semitendinosus, yeah. You can add in glutes. I would say that if it was a question on the test and you said glutes do hip extension, you wouldn't get it wrong. Yeah. Okay. And then knee flexors, these are also going to be hamstrings, right? So a little bit of knee flexion being caused by the hamstrings, and that gastrocnemius, which is a calf muscle. Remember, it goes up over the back of the knee a little bit too. Yeah. Okay, so that's for the push off part. And then we want to flex our hip up, right? What's our strongest hip flexor muscle? So one with a funny name? Iliopsoas, there it is for sure. So the iliopsoas, but then we have rectus femoris, and we also have pectineus to help us with that. And then what are our knee extensors? All the quads, right? Quadriceps femoris. Those are our knee extensors, all four of them. Right, so we got vastus medialis, vastus intermedius, vastus lateralis, and rectus femoris. Okay, and then ankle dorsiflexion. So this would be our tibialis anterior. That's our primary ankle dorsiflexor, but then also the one that helps us pull up our toes, which in turn helps us pull up our feet, is our extensor digitorum longus. Those are just kind of selected out of the ones that you need to know. There's other muscles that do some of these things, but not all ones that you need to know for this class. Okay. All right. Well, that's that. We're done. Have a great day, guys. We'll see you on Wednesday.
Cause I know that like, this is infraspinatus, this is like subscapular. But then how is this subscapular?
This one is looking like this. This one is looking like this. This one's on the back.
Okay, so it's like, so this is like the back.
And this one's the armpit.
Oh, and this is from the armpit.
Yeah.
And then for infraspinatus, just to like make it like, that like little slip.
Certainly, so questions aren't on the video.